Hello, my name is Jenny and I'm a molecular ecologist at the UK Centre for Ecology and Hydrology. I took over running of the National Honey Monitoring Scheme in October 2022. In this presentation, I'll be taking you behind the scenes of the scheme and talking a bit about the approaches in molecular biology involved in the processing the honey samples we ask beekeepers to send in. I'm focusing on this aspect of the scheme because participants quite often ask why it takes us a while to get their results back to them after sending in their samples. And I wonder if there's an assumption that there is a piece of machinery that we load the honey into that produces all the results we want overnight. The analysis is much more involved and time consuming than this, so that is what I'm going to talk you through. If you have any questions, please comment below the video and I'll do my best to answer them. I'm assuming you are already familiar with the scheme if you're watching this talk, so I'm only going to give a brief introduction. But if you have not heard of the scheme before, please visit our website www.honey-monitoring.ac.uk or follow our Twitter account at CEH_honey. Scientific projects are rarely the work of a single person, so before I continue, I'd like to draw your attention to the list of people on this slide who have been involved in the scheme, past or present. In particular, I would like to thank Richard Pywell for bringing in the funding for the scheme, Anna Oliver for setting it up in 2018 and running it so amazingly well for the first 4 years, Ben Woodcock for data analysis, results and presentations, and Eleanor Grove, who is currently processing the 2022 samples with me. So a brief introduction. What is the National Honey Monitoring Scheme? NHMS is a citizen science project asking beekeepers from all over the UK to send in honey samples that are collected directly from a single hive. We ask participants to collect honey into 3 30ml tubes, one for pollen analysis, one for pesticide analysis, and a third for our honey archive. The pollen analysis allows us to see what honeybees from that hive have been foraging on and how land use and agriculture in the area surrounding the hive influences this. The pesticide analysis shows us what pesticides honeybees are being exposed to while they are foraging. Our archive is a repository of all samples collected across all years to enable future research. This could be on, for example, honeybee diseases, antimicrobial resistance genes, or something in the environment that it hasn't occurred to us to monitor yet. We believe NHMS is the largest scale environmental DNA citizen science project in the world. NHMS was set up in 2018 in consultation with the BBKA and BFA. So far, around 3,000 beekeepers have taken part from England, Wales, Scotland and Northern Ireland. Already the team have published the honey processing pipeline and 2 papers looking at pesticide residues in the honey. In 2018, when the scheme was set up, it had 200 beekeepers take part, and the scheme has grown considerably since this time. 586 samples were sent in for 2019, 1,109 for 2020, 1,164 for 2021, and last year in 2022 we received nearly 1,500 samples. So this plot along the bottom shows you what regions of England, Wales and Scotland these samples have been sent in from. The most of the samples come from the southeast of England, followed by the southwest and the east of England, and we're always happy to receive more samples from other parts of the UK. This is an overview of the scheme's honey processing pipeline that I'm going to talk about in more detail over the next few slides. Briefly, our beekeepers collect honey directly from their hives and send it to us at CEH Wallingford. We extract DNA from the pollen grains that are in the honey, and then we amplify a region of this plant DNA known as a DNA barcode. These barcodes are sequenced on a next-generation sequencing platform, and bioinformatics are used to assign these barcodes back to the samples they were found in and to match them to the plant they came from. These results are then fed into the scheme's online systems and sent back to participating beekeepers. To start with, beekeepers who wish to take part create an account on the NHMS website and then request a sample pack. At this point, they just need to provide the location of the hive they intend to collect the samples from and how long the hive has been at this location. When we receive a sample pack request, we make up a sampling pack to send to the participant. This pack includes a letter with instructions for collecting the honey, 3 tubes, a Ziploc bag in case the honey leaks, and a free post return envelope. We ask that the honey is collected from the same hive at the same time, and that it is the most recently capped honey collected directly from the comb. This is because we really want to see a snapshot in time from an individual hive, not the mixed honey in an extracted sample. We ask that you ensure that the location is accurate, and this can be updated via your Taking Part page. Once a beekeeper has requested a sample pack, their online account will show this survey. Only the date on which the sample was collected is compulsory, but we really appreciate it if participants are able to fill in the remainder of the survey with additional information about the hive's health and productivity, as this allows us to do more analysis with the honey samples. This is a photo of our honey archive room. On the left are all the archived samples from previous years of the scheme, and on the right are all of the samples that were sent in 2022 that we are currently processing for pollen analysis. When we unpack the 3 tubes of honey from the return envelope, we label them all with a unique code. The 3 tubes you see here are labelled HME for Honey Monitoring E, E indicating the 5th year of the scheme, and they were the 737th envelope received in 2022. As I mentioned earlier, 1 sample is for pollen analysis, 1 is for pesticide analysis, and 1 is for the long-term archive. We weigh out 15 grams of the sample intended for pollen analysis and also apply this sample to the refractometer to measure the sugar and water content of the honey. These results are added to our website, and the beekeeper should receive an email notification that these results are available. The timescale takes us about an hour to process 10 samples at this stage. The aim of the filtering step is to capture the pollen grains that are suspended in the honey sample. The tube containing the 15 grams of honey from the previous step is topped up to 50 ml with sterile water and heated for an hour at 55 degrees C, with regular mixing to ensure the honey and wax are thoroughly suspended in the water. After an hour of heating, we remove the wax that has floated to the top of the tube and we vacuum filter it. This involves placing a clean filter paper onto a sterile filtering platform with a clean filter cup above it, pouring the honey sample into the cup, and then applying a vacuum to this cup. When the vacuum is applied, the honey is drawn through the filter paper, but the pollen grains are captured on top. This photo shows a filter paper coated in pollen after filtering compared to a clean filter paper. The filter paper is then folded into 8 using tweezers, placed in a cryotube, and stored in a -80 degrees C freezer. For a time scale, it takes a day to process 48 samples. So at this point, we have pollen grains on filter paper. Pollen is designed to be super resilient. It has to survive UV radiation, temperature extremes, buffeting, and ingestion in the environment to serve its purpose, which is to pollinate other plants of the same species. I found these tweets by Dr. Jacqueline Gill, a paleoecologist at the University of Maine, and I think she does a great roundup of how resilient pollen is. Firstly, pollen is made of sporopollenin, the most resilient natural biopolymer known to science. You can pull out pollen grains from hard rock dating back tens of millions of years. Pollen is an amazing forensic tool, helping us to reconstruct not just crime scenes today, like in CSI or Bones, but to tell the stories of the planet's ecosystems and civilizations in the past. And those stories provide clues to climate future. Did I mention how resilient pollen is? You can douse it with concentrated sulfuric acid and hydrofluoric acid, the stuff that dissolves sand, and it doesn't even care. To close, pollen is a versatile tool in paleoecology and archaeology. pollen is beautiful, resilient, and powerful. It's also super cool. It's the plant equivalent of identifying a giraffe from a lizard using only sperm over thousands or millions of years. Here comes the first tricky bit of the process. The goal here is to get DNA from those exceptionally tough pollen grains that have evolved to resist every damage mechanism nature can throw at them. To do this, we use a multi-pronged attack. Firstly, we use a chemical concoction that aims to break open the cells by changing the pH around them. This concoction also contains an enzyme, which breaks down proteins in the cell that would otherwise destroy the DNA we're trying to get to. Then for pollen grains that have resisted this chemical lysis, which is the term for breaking apart cells, we use mechanical lysis. We put them in a tube with tungsten carbide beads, tungsten being the strongest metal in the world, and then put them in a machine that shakes the tube incredibly violently and pulverizes everything within it. At this point, we have what's called a lysate, which contains DNA, RNA, proteins, cell debris, and other things that were collected on the filter paper during the filtering stage. We only want the DNA. The other cell components can hinder the next stage of processing. We load this lysate into what's called a spin column, which contains a different kind of filter called a matrix that only binds DNA. Multiple wash buffers are added to the spin column and flushed through, taking everything through with them apart from the DNA, which is left bound to the matrix in the spin column. When all the contaminants are washed through, we add an elution buffer, which releases the DNA that is bound to the matrix, and this elution is then stored at -20 degrees C. In terms of timing, this step represents a bottleneck to the processing pipeline, as we can only process 24 samples a day. Just like when you go into a supermarket and scan the barcode, which is unique for an item, every living organism has its own barcode in its DNA that we can use to identify it. DNA barcodes are a fragment of DNA and exist for animals, plants, bacteria, and fungi. From the DNA extraction process I talked about in the last slide, we have all of the DNA that was extracted from each pollen grain on the filter. And in this step, we want to hone in on one piece of DNA that works as the plant's barcode. To do this, we add what are called primers, short pieces of DNA that bind either side of the DNA barcode. And then we perform a PCR, which is hopefully not an unfamiliar term now, thanks to COVID. And this creates thousands of copies of the DNA barcode bounded by these primers. In this way, we have far more copies of the DNA barcode than of the rest of the DNA, which enables us to sequence just the DNA barcode. Timing-wise, it takes about 3 hours at this point to process 96 samples. This is a nerdy slide because I still find this process fascinating, and it's been in use since the 1970s. So if you have done any genetics-based lab work, you will know what this is. The last 2 slides have shown DNA extractions and multiplication of the DNA barcode, and these are both really finicky processes that often don't work. However, you won't know that they've not worked until you get to this point. These pictures are of a process called gel electrophoresis. We make a gel, which is akin to a thin layer of jelly with indents at the top for us to pipette our samples into. This gel contains a fluorescent dye, which binds to DNA. So when you add your samples into the top of the gel, any DNA present will bind to this fluorescent dye. Then an electric current is passed through the gel from top to bottom, and because DNA has a negative charge, it will move through the gel in the direction of the current. Small pieces of DNA will move faster than larger pieces, so after time you will get a laddering effect of DNA throughout the gel. When you shine a UV light on the gel, any DNA present will fluoresce, and you will be able to tell the size of your DNA pieces by where they are placed in the gel. The photo on the left shows DNA fluorescing under a UV light, and the photo on the right shows plant DNA barcodes that have been multiplied from pollen DNA. This way, you can see A, if your DNA extractions and PCRs have been successful, and B, if the DNA pieces are the correct size for the DNA barcode you are using. When we sequence a sample to find out what DNA barcodes it contains, we do not do this for each sample separately. We add codes to the beginning and end of the DNA barcodes. that are a unique combination for each sample, and then we pull multiple samples together. There are so many combinations of these codes that we're able to pull 400 samples into one for sequencing. Sequencing is done using a MiSeq machine and produces pages and pages of DNA sequences that look like this, all made up of 4 letters A, T, C, and G. We can then use the code combination to assign the DNA sequences back to the sample they came from. For timescale, it takes about a week to do 394 samples at this stage. By comparing the DNA sequences to databases of plant DNA, we're able to identify which plant the pollen grains came from. This image shows an example DNA database for a single plant that has had multiple plant DNA barcodes sequenced. You can see here at the top the DNA barcode called ITS2, which is the barcode we use in the honey monitoring scheme. This slide shows a top-down of taxonomic identification, using brambles as an example. The top level of the taxonomy is kingdom, which for plants includes about 280,000 species. The second level is division, which for flowering plants contains about 250,000 species. The order Rosales contains about 18,000 species, and the family Rosacea contains about 3,500 species. It's at this point that it gets trickier because assigning genus or species relies on the DNA barcode being significantly different between species to be able to tell them apart. For brambles, there are between 250 and 700 species within the genus Rubus, and these have very similar DNA barcodes. So it is not possible to tell apart, for example, blackberries, raspberries, loganberries, and tayberries from their DNA barcodes alone. This challenge also applies to brassicas because of how closely related the most common brassica species are. Brassica oleracea is a species that includes the common cultivars kale, cabbage, kohlrabi, cauliflower, broccoli, and Brussels sprouts. Crossed with Brassica rapa, which is turnip or Chinese cabbage, it gives oilseed rape, and crossed with black mustard, it gives Ethiopian mustard. This means oilseed rape is sometimes misclassified as turnip, for example, because their DNA barcodes are so similar. Once all analyses are complete, each beekeeper is sent a report for the hive they collected the samples from, which provides a bar chart showing the top 15 plant taxa present in that honey sample and a complete list of all of the plant taxa within that sample. Participants are also sent a land cover report that is generated using the UKCH land cover maps, which assign land cover and crop type to every parcel of land in the UK using satellite imagery. Land cover is generated for the 2-kilometre radius around the beehive that honey is sampled from, and 2 pie charts are produced, one showing crops surrounding the hive and the other showing habitat surrounding the hive. So just a little note at this point that the total cost per honey sample processed is around £100. I hope you found this talk interesting and that it's given you some insight into the processing pipeline for pollen analysis from honey. If you have any questions, please comment them below this video and I'll do my best to answer them. And also, if you have any ideas of what research we could do on the archived honey samples, please get in touch. Thank you for your interest in the scheme and for your support if you have previously sent in samples, and thank you for listening.